The first ever full genome analysis of ancient Egyptians shows they were more Turkish and European than African. Scientists analyzed ancient DNA from Egyptian mummies, dating from 1400 BC to 400 AD, and discovered they shared genes with people from the Mediterranean. They found that ancient Egyptians were closely related to ancient populations in the Levant, now modern-day Turkey, Syria, Jordan, Israel and Lebanon. They were also genetically similar to Neolithic populations from the Anatolian Peninsula and Europe. The groundbreaking study used recent advances in DNA sequencing techniques to undertake a closer examination of mummy genetics than ever before. The study, published in Nature Communications, found that modern Egyptians share more ancestry with Sub-Saharan Africans than ancient Egyptians did. The data shows that modern Egyptians share approximately 8% more ancestry on the nuclear level with Sub-Saharan African populations than with ancient Egyptians. Egypt is a promising location for the study of ancient populations because it was a worldwide trading hub. This is likely the reason that ancient Egyptians had such a diverse genetic heritage, the authors, from the University of Tübingen and the Max Planck Institute for the Science of Human History and Gene, said. The population history of Egypt is complex because it is found at the Isthmus of Africa, the gateway to a continent, and has seen much historical turnover. Max Planck Director for the Science of Human History and Study Lead Author Professor Johannes Kraus told Mail Online. Ancient Egypt in the 1 millennium BC had been dominated by many foreign powers. The team's research involved unraveling the genetic history of Egyptians by comparing DNA samples taken from both modern and ancient natives. The researchers were aiming to establish an exhaustive genetic database to study the ancient past of Egypt for the first time. It has been much debated whether foreign dominations such as Assyrians, Nubians, Greeks or Romans changed the gene pool of ancient Europe, making them more or less African, Professor Kraus told Mail Online. We wanted to test that, and found that there is genetic continuity between the Old Kingdom and Roman period. However, in the last 1,500 years Egypt became more genetically African, whereas the ancient Egyptians showed almost no Sub-Saharan African ancestry, and high affinity to ancient Near Eastern and European populations. The team sampled 151 mummified individuals from the archaeological site of el-Meleq, along the Nile River in Middle Egypt. Recent advances in the study of ancient DNA present an intriguing opportunity to test existing understandings of Egyptian history using the ancient genetic data. The new study managed to extract accurate, full-genome DNA data from 3 ancient Egyptian mummies, and usable segments of DNA from 90 mummies. The team used next-generation sequencing methods to read stretches of any DNA present in a sample, and retrieve those that resembled human DNA. The complete reads allowed the team to spot telltale damage patterns associated only with ancient DNA. This makes the new study's results much more reliable than those of any mummy DNA research that has come before. The extraction of reliable nuclear DNA from Egyptian mummies is hence a breakthrough in genetics that opens the door to more detailed studies of mummified remains. They were able to use the data gathered to test previous hypotheses drawn from archaeological and historical data and from studies of modern DNA. Professor Alexander Peltzer, from the University of Tübingen, said, "In particular, we were interested in looking at changes and continuities in the genetic makeup of the ancient inhabitants of Abuser el-Meleq. We wanted to test if the conquest of Alexander the Great and other foreign powers has left a genetic imprint on the ancient Egyptian population." The team wanted to determine if the investigated ancient populations were affected at the genetic level by foreign conquest and domination during the time period under study, and compared these populations to modern Egyptian comparative populations. The study found that ancient Egyptians were most closely related to ancient populations in the Levant, modern-day Turkey, Syria, Jordan, Israel and Lebanon, and were also closely related to Neolithic populations from the Anatolian Peninsula and Europe. Co-author Wolfgang Hauck, group leader at the Max Planck Institute, added, "The genetics of the Abuser el-Meleq community did not undergo any major shifts during the 1,300-year time span we studied, suggesting that the population remained genetically relatively unaffected by foreign conquest and rule."